I am Subhajit Khan and I am currently a final year MSc Life Science student at NIT Rourkela. I am starting my YouTube journey with this first video and I hope you would like it and please share your views. As I said, I am a MSc final year Life Science student at NIT Rourkela. I just finished my summer internship at IIT Kharagpur in Department of Biotechnology and Biosciences in Molecular Biology and Immunotechnology lab. And I have secured All India rank 66 in IIT JAM in 2023 and All India rank 222 in GATE BT this year only. And some more details about me is mentioned here. And I did my graduation from University of Calcutta in microbiology honors, which also included chemistry and botany as my general subjects, and I secured grade A+. My favorite subjects which I like to teach is biology and chemistry and till 10+2 level, and I can teach microbiology and biochemistry till graduation level also. So now today in this video we'll be focusing on carbohydrates, which is a part of biomolecules. It is an important topic in IIT JEE. Now to understand what are carbohydrates, we first need to understand what are biomolecules. Now living system is made up of complex biomolecules such as carbohydrates, proteins, enzymes, lipids, vitamins, hormones and nucleic acids. Most of the biochemical reaction takes place at pH 7, 37 degrees Celsius, which is our body temperature, and at 1 atmospheric pressure. So all these are involved in biomolecules, but in this video we will be focusing on carbohydrates only. Now let's come to classification of carbohydrates. Carbohydrates can be classified into monosaccharides, oligosaccharides and polysaccharides. Now monosaccharides are those in which they cannot be further hydrolyzed into polyhydroxyaldehydes or ketones, such as there are actually 20 natural monosaccharides known till now. Some of them are glucose, fructose, ribose, etc. Now oligosaccharides are those which can be hydrolyzed and they form 2 to 10 monosaccharides on hydrolysis. Some of them are sucrose, maltose, raffinose, stachyose. Now oligosaccharides can be further classified into disaccharides, trisaccharides and so on, depending on how much monomeric units they would be hydrolyzed into. Now another is polysaccharides, which are very high molecular weight carbohydrates, and these contains large number of monosaccharides. These form large number of monosaccharides on hydrolysis. Example, starch, cellulose, etc. Now below we can see different examples of monosaccharides. Now when we compare these three, glucose, mannose and galactose, we can see something at the between glucose and mannose. When we number it, the carbon atoms 1, 2, 3, 4, 5 and 6 and 1, 2, 3, 4, 5, 6. You can notice that both glucose and mannose are nearly similar. There is one difference at the C2 carbon atom. The positions of both the Fischer projections are almost same. Just in the C2 carbon atom, the OH is on the right side, and in mannose, in glucose, and in mannose the C2 OH is on the left side. Now this difference is called C2 epimer. Now when we say about epimers, epimers are those diastereomers which differ in only a single carbon atom in their configuration. They are diastereomers because their formula are same, just their geometry is different, but they differ only a single carbon atom, that is at C2 position. So since the difference is only at C2 position, so glucose and mannose are C2 epimers of each other, or mannose is a C2 epimer of glucose. Similarly, when we compare glucose with galactose, you can see this is also an epimer. You can tell which carbon atom it is an epimer of glucose. It is 1, 2, 3, 4, 5. We can see at the 4th carbon atom, the OH is on the left side of galactose, and in glucose it is on the right side. So from here we can say that This is a C4 epimer. Both glucose and galactose are diastereomers. They have same molecular formula, and just they differ in their geometry. So when we see that the C4, there is only a difference in geometry in only single carbon atom, that is at C4. So they are C4 epimers. So galactose is a C4 epimer of glucose. Mannose is a C2 epimer of glucose. Now, when we say these are aldohexose, glucose, mannose and galactose. These are aldohexose. Since there we can see an aldehyde group, and there are 6 carbon atoms. So it is an aldohexose. While when we see about fructose, we can see at the C2 carbon atom, it is a keto group instead of aldehyde group at the C1 position like others. Fructose has a keto group at the second carbon atom. So it is a hexose due to 6 carbon atom. So fructose is a ketohexose. So let's move on to our next topic. This is a PYQ. I hope you would answer it. We have just read it. So the question is, fructose is an example of. So quickly say, you can write in comments also. So what will be the answer? It shouldn't take much time. The answer is ketohexose. Okay. Now, when we come to carbohydrates, we generally refer it as hydrates of carbon having the general formula CxH2O whole Y. But there are many exceptions to it, such as when we talk about ethanoic acid, it is not a carbohydrate, but it has the formula CH3COOH, which follows the general formula, but it is not a carbohydrate, it is an acid. So another exception is the rhamnose, as I already told. It has the formula C6H12O5, but it is a sugar, it is a carbohydrate, but it does not follow the general formula. Again, another example which is an exception to the general formula is D2-deoxyribose. It also does not follow the general formula, as when we see 1, 2, 3, 4 and 5. In the C2 carbon we can see there is no OH group. Both sides there are H and H. So when we see the formula, it does not follow this formula. There is one H2, second H2, but another H2 is missing. So we cannot it is a carbohydrate, but it does not follow the general formula. So what is the actual definition? As told, it should be optically active. It should be polyhydroxyaldehydes or ketones or substances that would yield this type of compound on hydrolysis. So when we see D2-deoxyribose, the second carbon is also chiral, the third carbon is also chiral, and even the fourth carbon is also chiral. Now chiral carbon means on the 4 hands of the carbon there are different groups. So there are 3 chiral carbon atoms. So it is an optically active compound in this sense. Also, when we see polyhydroxyaldehyde, here CHO, it is an aldehyde group. So it follows the definition of carbohydrates. Similar with the D-rhamnose, the C1, C2, C3, C4, C5, C6. In we can see the C2, C3, C4 and C5 all are chiral carbons. And also with an aldehyde group at the top. So it is also a carbohydrate. Now we can say about D-ribose, it is also a carbohydrate and it also follows the formula. So most of the carbohydrates follows this formula. There are a few exceptions as mentioned. But most of the carbohydrates follows the general formula. The few exceptions are mentioned here. Now we'll be discussing our 3 important things, that are what are sugars and non-sugars, what are reducing sugars and non-reducing sugars, and what is DL system. Now sugars includes all the monosaccharides and oligosaccharides because they are mostly crystalline, soluble in water and sweet. So sugars are crystalline, soluble in water and are sweet, and they includes all the monosaccharides and oligosaccharides. Now The non-sugars includes polysaccharides as they are amorphous, insoluble in water, and are tasteless. So, now sugars can be reducing as well as non-reducing. The reducing sugars are those that contains a free aldehyde group or a free ketone group, so that they can reduce the Felling's solution or the Tollens reagent. So if they can reduce the Felling's solution or the Tollens reagent and contains because they contain a free aldehydic or ketone group, so they are called reducing sugars. All monosaccharides are reducing sugars, and all disaccharides are reducing sugars, but in disaccharides there is an exception, that is sucrose. Except sucrose, all disaccharides are reducing sugars. Sucrose is not a reducing sugar because both the glucose and galactose, the reducing group, are joined together to form sucrose. So sucrose as a disaccharide is not a reducing sugar. Now let's come to the DL system. Before DL system, you should understand that it is different from the plus and minus system of the stereochemistry, because the plus system denotes the dextrorotatory molecule and the minus denotes the levorotatory molecule. So do not confuse it with the DL system. It is often confused. So now what is the DL system actually? In DL system, we consider the lowest asymmetric carbon atom of the molecule in the Fischer projection. Now it is generally the reference is generally taken as glyceraldehyde for carbohydrates. Now in carbohydrates, what is the reference is that the OH group, if the OH group is towards the left of the lowest asymmetric carbon atom, then it is called a L. Since here the OH group is at the left, it is called L-glyceraldehyde, and minus for levorotatory. So if the OH group is towards the right of the lowest asymmetric carbon atom, then it is called D-glyceraldehyde. Similarly, mostly glyceraldehyde is taken as a reference because it has only one lowest asymmetric carbon atom. So similarly, when it comes to larger molecules such as a hexose like D-glucose, we take the lowest carbon atom, which is the C5 carbon atom. In the C5 position, we can see the OH group is towards the right, so it is a D-glucose. So in this way, we can name it as D or L configuration. Now let's come to an important topic, that is cyclic structure of glucose and fructose. Now before doing that, we first need to know what is alpha configuration and what is beta configuration, just like we learned D and L configuration. Now previously when we hear that D or L configuration, now seeing this, what do you think? It is D or L configuration? Since the OH is of the lowest asymmetric carbon atom is towards the right, so it is D configuration. So when we can see here, it is OH is towards the right in both the cases, so it is in D configuration. Now what is alpha and beta configuration? In the Fischer projection, if the OH group is towards the right, alpha is always right. Generally I don't support that, but we heard that sigma, like sigma male, that's we call generally they consider it right, but I don't support that actually. But there was something, so we can say just like that, alpha is always right. So alpha, in case of alpha, the OH group is at the right position in the Fischer projection, and in the beta, the OH group is towards the left, and this is considered for the C1 carbon atom in the Fischer projection. So if the OH group is towards the right, it is alpha, and if the OH group is towards the left, it is beta. Now in Fischer projection also, it is considered when the lowest asymmetric carbon atom, the OH group of the lowest asymmetric carbon atom, attacks the C1 carbon atom. So here what happens is that oxygen contains lone pairs. These lone pairs attacks the C1 carbon atom to form a ring-like structure. We can see here a ether-like bond is forming here. between the C1 carbon atom and the C5 carbon atom. So this ether-like bond causes this Fischer projection structure to form a ring-like structure. Since the C5 is connected to the C1 carbon atom, it forms a structure similar to the pyran ring, which is a 6-membered ring structure. So when this structure is formed in a cyclic structure, it is called alpha D glucopyranose. Now, how to understand it in this ring configuration? What is alpha and what is beta? Generally, in Hindi, beta means beta. Beta is considered upward generally. So, in this case, in beta configuration, the OH group is present above the plane. So here, since the OH group of the C1 carbon atom is present above the plane, we consider it as a beta configuration in the pyranose form, pyranose or in furanose or in the ring structure. In case of alpha, the OH group is below the plane. So when the OH group of the C1 carbon atom is below the plane, it is considered as alpha configuration, and when the OH group is above the plane, it is called beta configuration. So when the ring formation occurs, there are two structures we get: alpha D plus glucopyranose and beta D plus glucopyranose. Depending on the C1, the OH group of the C1 carbon atom is above the plane or below the plane. Now similarly, it goes for fructose. Now in fructose, we can get two structures. Since we know that the C1 carbon atom, C2 carbon atom is a keto group. So in case of glucose, the C1 is was the aldehyde group. But in case of fructose, the C2 carbon is the keto group, and that oxygen of the keto group binds to the C5 carbon atom. Means the oxygen atom gives its lone pair to the C2 carbon atom to form a ether-like bond, and this causes a tension in the structure to form a ring-like configuration, that is fructofuranose. Now you can see and tell it if it is alpha configuration, beta configuration, and is it D or L configuration. So seeing this Fischer projection, we can say that it is a D configuration, since the OH is towards the oxygen is towards the right, and in alpha it should be alpha because the OH group is below the plane. So we can say it is alpha D fructofuranose. Similarly, if we want to show it as beta, we can remove this OH group and we can say OH and CH2OH, and then it would be in beta D fructofuranose. So let's move to next topic. Now let's end today's lecture with a PYQ. You can pause. You can see this question, answer it by pausing it for 1 minute. Let me solve this for you. The question says the correct representation in 6-membered pyranose form of the following sugar X is. Given this sugar this, and there are 4 options. So which one is the correct option? Ring form. You have to answer it. So when we see this, number it. First number it. 4, 5, 6. So and in the ring form. 6. So we can see here that it should be the things which are on the right should be below the plane in the ring, and which is on the left should be above the plane. So we can see here that only in option B, the 2, 3, the OH group is on the left, so it is above the plane, and when in 4, 5, the 5 oxygen, the lone pair will be donated to the first carbon, so it should not have any oxygen. So the correct option should be option B. I hope you answered it right. Thank you.